Good afternoon, everyone. I'm Joaquim Tomàs, and I'll present to you about our work on Bayesian estimation of DEB parameters using STAN, conducted alongside with Miquel Pelmé and Andrea Campos. I will start the introduction with the relevance of interindividual variability. At the eco-evolutionary level, it is relevant to estimate this variability in different DEB parameters with certain accuracy and precision, rather than focus only at the population level. Focusing on each individual means that we cannot assume that they are clones. Each one has its singularities. Henceforth, its possible differences between individuals can help us to understand many processes at population level. We aim in a shift in aquaculture, with management based in processes derived from DEB models relevant at individual level, for example, optimization of food assimilation, instead of conventionally used covariation patterns as weighted age. Therefore, we want to implement the mechanistic approach of DEB models to aquaculture with a focus on interindividual variability. DEB parameters estimation is still a challenge despite the improvements in the calculation methods, as DEB tools. The vast number of parameters to estimate in a model is one of the main difficulties that limit the expansion of the DEB applications. Moreover, to estimate parameters at the individual level implies more complexity. The main goal of this study is to develop an implementation with the STAN software capable of estimate the interindividual variability in the parameters of a particular fish species. We have focused in estimating variability in 4 specific parameters: the kappa fraction, the energetic conductance, B dot, the maximum assimilation flux, PIM, and the volume-specific somatic maintenance rate, Km. The aim of this talk is to show the steps followed to estimate them with STAN, as well as the limitations we have found through the process. Why do we have decided to use STAN? There have been several revisions to cope with the limitations of parameter estimation in DEB, as the Lika covariation method, as well as the improvements in the loss functions implemented in the DEB tools and its revisions. However, between individual variability and the estimates uncertainty are hardly ever considered. This is why the use of Bayesian inference and hierarchical mixed models is expanding, in order to cope the 2 problems mentioned before. Those methods are supported by several authors as Johnson and Bersupan. The need for a system of numerical resolution of coupled differential equations at each iteration can be a computational handicap. STAN has an integrated function for numerical integration, which offers a complete Bayesian inference by using more efficient algorithms. From simulation to estimation. First, we started simulating realistic data with interindividual variability for the fish species Serranus scriba, using the DEB parameters from the species entry in AtMyPet. We have used the standard DEB model with temperature and food as forcing variables, and the simulated dataset for the observable variables, fish length at age, and body and gonads wet weight. Then we took this emulated realistic dataset, in which we proceeded to perform a longitudinal sampling, creating a dataset with 10 simulated fishes, measured annually through an 11 years lifespan. In order to make our simulated data more realistic, we introduced a variability source to emulate the measurement error. Afterwards, we performed the parameter estimation with STAN and compared them with the 2 parameter values in order to obtain the accuracy and precision of our estimates. We will present our process to obtain the results, showing our exploration process from the starting point, as well as the problems we have encountered midways. At the beginning, we started analyzing only one fish, in which we had 11 years of annual measurements of length and wet body and gonads weight. We started estimating only one parameter, PIM. First, we estimated it using only length data, and then using the 3 observed variables. Here you can see the plots with the results of the PIM estimations. In red, the density of the estimates, with the dashed lines representing the 95% credibility interval, and in black, the true value of the parameter. Up until now, with a simple preliminary analysis with one fish and one parameter to estimate, the resulting estimates are pretty accurate and precise. We can see how the parameter estimations improved when we added more data input to the model. We repeated the same procedure fish by fish and we got similar patterns for the estimations. The next step was to estimate PIM along with kappa and V dot. In the plots, we can see the results for the three estimates distribution density. The accuracy and precision of the PIM estimation remains quite good, and the new estimations for kappa and V dot are acceptable too. We performed a pairs plot in order to verify that there is not any possible correlation between the three estimates. The plot shows us the comparisons between the estimates of each parameter and their true values, which are represented by the blue lines. The scatter plot shows the density of the estimates, and histograms represent its posterior distribution. The contour plots point to the credibility intervals of the estimates. We can observe as the estimates have been obtained with high precision and satisfactory accuracy, and in addition it seems that there is not any correlation between parameter estimates. So we carried on adding complexity by performing an analysis considering 4 parameters to estimate: the previous 3 and the somatic maintenance rate, Km. Now you can see the same plot as before, but with the estimations for the 4 parameters. Here we can see one of the major problems we have encountered in our analysis. There are 2 strong correlating parameters, PIM and Km. This involves a major handicap in our estimation process, as we cannot estimate separately these 2 parameters with the given conditions. Due to their correlation, different combinations of values can lead to similar results. Although generally the estimation's accuracy has improved, the new estimates now turn PIM to be less precise. As we could not separate PIM and Km with the 3 given observed variables, we entered a new observed variable, oxygen consumption. And we performed a new analysis, but the results did not improve. The parameters PIM and Km kept their correlation. Adding this new observed variable did not imply any improvement in our estimations, and moreover, it did not solve our problem with the parameter correlation. Here we made our first step back. We kept analyzing one fish and the 3 initial observed variables and 4 parameters, as in the third scenario, but adding variability to the functional response. We converted the functional response, a fixed parameter until now, into a parameter with a sinusoidal variability. But after obtaining the parameter estimates, we observed that adding variability to the functional response did not help either to solve this problem. Thus, we took a step back to the second scenario, but now we will analyze 10 fishes at once. So, we estimated the 3 parameters, PIM, kappa, and V dot, leaving Km, assuming it cannot be separated from PIM. And now, as we had more than one individual, we assumed that the between individual variability of the parameters was distributed along a normal distribution for PIM and V dot, and logically distributed in the case of kappa. In the new simulations, each parameter was allowed to vary 20% between individuals over a median population average value, based in the reports for between population variability of Potter et al. For each fish, we obtained growth curves of length, body weight, and accumulated fecundity. We obtained the estimates for each parameter for each fish, and then we compared them with the true values. As we can see in the plots, the dashed line indicates a perfect estimation of the value, and each dot represents the parameter of one fish and its 95% credibility interval. You can observe how PIM is estimated with high accuracy and precision, while kappa has an acceptable accuracy and precision, but the estimates of B dot have low precision. We also performed estimations of the measurement errors for each observed variable. We were able to obtain accurate estimates from the measurement error of the body length and the body wet weight, but not for the accumulated fecundity that is overestimated. At this point, as the last analysis, we experimented on what will happen if we increased the variability of the DEB parameters from 20% to 30% in the case of kappa and B dot, but we kept the PIM variability remaining the same at 20%, as increasing it could cause huge differences on the observed variables. We can observe now how the accuracy and precision of the estimates improves when there is more between fish variability of the DEB parameters. After all those analyses, we can jump on some conclusions at different levels. The precision of the parameter estimates of PIM, kappa, and B dot obtained with STAN was quite good when considering there's 3 observable variables. Some pairs of DEB parameters may remain structurally non-identifiable because different combinations of values can result in virtually the same observed patterns. With the considered model, we were not able to separate PIM and KM. Adding oxygen consumption as a variable or functional response variability did not suppose a clear advantage on parameter estimation. Measurement errors were also able to estimate well, except for the accumulated fecundity, in which we suspect that there was an error in the code. Simulation analysis, as the one presented here, may help in finding the proper set of observable variables and the optimal remonitoring time needed for properly identifying some DEB parameters. STAN proved to be an appropriate tool to retrieve quite good individual parameter estimates with an acceptable accuracy and precision. Thus, is a promising tool for aquaculture, where knowing the DEB parameters at individual level opens a wide range of applied research opportunities. We must mention that we have encountered an exponential increase in the computational time when running more than one individual at the same time. Analyzing many fishes, each one having many observed variables and repeated observations, considering many parameters, each additional factor increases exponentially the computation time. For further steps, we plan new analyses to identify proper datasets of observed variables and experimental designs that could allow better identification and estimation of the inter-individual variability for target DEB parameters that are relevant in our purpose in aquaculture. Our goal is this technical process is to understand the detected correlation patterns in parameter estimates and solve the handicap of not being able to separate them. Well, that's all. Thank you for your attention. Here attached you have my email address and for any questions about that work. Goodbye.